My name is Aditya Chatterjee, and my topic is microbiome and microbes in animal ruminant. So, what is human microbiome? Human microbiome is the full array of organisms that live on and in humans, and more specifically, the collection of microbial genomes that contribute to the broader genetic portrait or metagenome of a human. The genomes that constitute the human microbiomes represently remarkably diverse array of microorganisms that includes bacteria, archaea, fungi, and even some protozoans and non-living viruses. Bacteria are by far the most numerous members of the human microbiome. The bacterial population alone is estimated at between 75 trillion and 200 trillion individual organisms. The sheer microbial abundance suggests that the human body is in fact a supraorganism, a collection of human and microbial cells and genes, and thus a blend of human and microbial traits. So, why a healthy microbiome matters? Healthy microbiome keeps our gut healthy and protects our immune system and supports our immune system. Microbiome. The microbiome is a layer of organisms including friendly bacteria. Gut wall. Friendly bacteria in the microbiome protect the gut wall from the damage and help with everything from digestion to mood. Immune system. Friendly bacteria also helps to regulate and support the immune system. So let's talk about the discovery of the microbiome. The first scientific evidence that microbiomes are part of the normal human system emerged in the mid 1880s when Austrian pediatrician Theodor Escherich observed a type of bacteria, Escherichia coli, in the intestinal flora of healthy children and children affected by the diarrheal disease. The years followed, scientists described a number of other organisms isolated from the human bodies, which include Velonella parvula, a bacterial member of the oral, digestive, urinary, and upper respiratory flora, and in 1900, Bifidobacteria, members of the intestinal flora. In the 20th century, a number of other organisms were isolated from the nasal passages, oral cavities, skin, gastrointestinal tract, urogenital tract, etc. So, how microbiome helps in our human body, and what is the role of the microbiome in the human body? So, if we talk about nose, it helps in mucus production, antimicrobial chemicals. Mouth assist in digestion, ward of pathogens. Lungs lubricate pulmonary tissues. Stomach prevents gastric complications. Colon digestion of complex carbohydrates. Sexual organs maintain pH and H2O to production to kill microbes. Skin fortify immune system, scent production. There are some more roles of microbiome in human body. Population of microbes inhibit the skin and mucosal surfaces in various parts of the body. The role forms part of normal healthy human physiology. The Human Microbiome Project found that individual host thousands of bacterial types, different body sites having their own distinctive communities. Skin and vaginal size showed smaller diversity than the mouth and gut. These showing the greatest richness. It is estimated that 500 to 1000 species of bacteria live in the human gut but belong to just a few phyla. Firmicutes and bacteriotides dominate, but other there are proteobacteria, verochromia, actinobacteria, fusobacteria, cyanobacteria, which are present. The vaginal microflora consists mostly of various lactobacillus species. Many organisms in the human microbiota that are closely related to pathogenic. So many microbiomes cause diseases also. So we are going to talk about that. The bacterial species such as Staphylococcus, Streptococcus, Enterococcus, Klebsiella, Entobacter, and Neisseria are the pathogenic microbiomes. Clostridium difficile infection serves as a useful example for illustrating the significance of the relationship between the human microbiome and healthy and disease. C. difficile infection, which is characterized by severe recurrent diarrhea, abdominal cramping, and nausea. Now we are going to talk about human microbiome. Microbes in the reticulum include bacteria, protozoa, fungi, archaea, and viruses. Bacteria, along with protozoa, are the predominant microbes, and by mass count of 40 to 60% of total microbial matter in the rumen. They are characterized into several functional groups such as fibrolytic, amylolytic, and proteolytic. Protozoa derive their nutrients through the phagocytosis of microbes and degrade the digest, feed carbohydrates, especially starch and sugars, and protein. Although protozoa are not essential for human functioning, the presence has pronounced effects. Ruminal fungi make up only 5 to 10% of microbes and are absent on diets poor in fiber. Despite their low numbers, the fungi still occupy an important niche in human because they hydrolyze some ester linkages between lignin and hemicellulose or cellulose and help break down digester particles. Human archaea, almost 3% of total microbes, are mostly autotrophic methanogens, produce methane through anaerobic respiration. So, more about human microbiome. Microorganisms number function in the rumen, and I have divided the table into that. So in the bacterial count in rumen is 10 is to 10 cells per milliliter. Ferment degrade a range of substrates and reproduce. Protozoa, uh, 10 is to 5 to 10 is to 6 cells per ml. Ferment degrade substances and gulls starch, bacteria, and other particles. Archaea, 10 is to 7 to 10 is to 9 cells per ml, metabolize hydrogen. Fungi, 10 is to 3 to 10 is to 5 zoospores per ml. Source of cellulitic enzymes, fiber degradation and digestion. Bacteriophages, 10 is to 8 to 10 is to 9 cells per ml, infect bacteria. So this is all about human microbiome and human microbiome.